Metabolism is a set of chemical reactions that is intended to keep the body at equilibrium or homeostasis.
In order to study metabolism, you need to measure the metabolites themselves.
A metabolite is in fact a small molecule that is produced by living organisms. Plants are kings of producing small molecules. They produce thousands and thousands of them. Metabolites are in fact the notion of the plant. This is what makes a rose smell like a rose and a tomato taste like a tomato.
Metabolites are not only important in plant research, but they're also very useful as a biomarkers for different human disease.
Almost every metabolic pathway in our body is rhythmically regulated by the clock. Circadian clock, these are clocks that measure around the day, measure around 24 hours. There is a reciprocal relationship between circadian clocks and metabolism. The clock regulates all the enzymes that synthesize these different metabolites. Therefore, we do know that maintaining the clock functioning properly can serve as a very good way to prevent metabolic diseases, cancer, and even other type of diseases like depression.
We're looking at specific metabolic genes and specific metabolites, and we're trying to see if we manipulate their levels, are we able to influence the progression of cancer?
We specifically collect blood samples from children with depression and anxiety, and we try to predict using metabolites or metabolic profiling in the blood of this kid the chance that they will develop severe side effects due to the treatment.
We focused on colon cancer because it has a direct interaction with the diet and the nutrients, and we try to understand the metabolic interaction between the cancer cells and the environment in the colon, with the hope that if we understand this interaction, we can intervene either by recommending preventive therapy, by providing early detection.
One of the group of metabolites that we have been studying are pigments called betalains. For us, these molecules are really strong antioxidants. They are known as supplements that we can use to improve our health. One of the intermediates of this pathway that leads to the production of the pigments is L-DOPA. L-DOPA is a very important drug for Parkinson, and in fact now we have in hand the way to produce L-DOPA to very high levels and purity in other systems by engineering.
One example how we can use metabolites to intervene with the function of the circadian clock is a study that was conducted in my lab and related to a group of metabolites which are called polyamines. And in this study, we very nicely showed that the clock regulates the level of these metabolites, these polyamines, and these polyamines can affect the function of the clock. They actually determine the frequency of the oscillator and maintain it with the 24-hour rhythms. And this suggests that by nutritional intervention, giving these metabolites in the diet, we can keep the clock ticking properly and can be used in the future maybe for therapies that can age decline in clock function.
Before the era of mass spectrometry, people could only study a few molecules. What metabolomics allows us is to really look at hundreds and thousands of molecules in one go. Mass spectrometry is really the best tool we have today to detect and identify these molecules, reconstruct it back, and then understand what is the identity of these chemicals.
I think there is definitely need for a facility that will enable us to measure in a rapid and efficient way metabolites and in different organisms.
We, for example, collaborate with scientists in the field of nutrition and the field of personalized medicine. Learning how to engineer these metabolites is in fact the future of human health.